Okay, I think we should start now. Okay, so it's a pleasure to have Himani Sachdeva for this research seminar. So Himani is also a physicist by training. She did a PhD in statistical physics, working on certain non-equilibrium phase transitions. After that, she decided to switch to population genetics, and she worked. She has done a lot of nice work with Nick Barton, and now she is at the University of Vienna. And today she'll tell us about polygenic local adaptation. Go ahead, Himani.
Okay, thanks, Kavita. So thanks for the invitation. A broad aim that motivates a lot of research in evolutionary biology is to understand how populations adapt to environmental conditions that vary over space and time. And when adaptation to one's own environment, that is local adaptation, might lead to reproductive isolation and speciation. And by reproductive isolation, we simply mean that there's a sharp reduction in gene flow or mixing between populations. And if we sort of take a genic view of speciation, then this sharp reduction really takes place mixing takes place all over the genome. And we would like to understand when local adaptation actually results in this sort of scenario. So the picture that we might have is of an extended population, maybe a patchy one consisting of many small subpopulations, which are denoted by circles on this schematic. And these subpopulations may be connected to each other in some complex way. Different subpopulations might also experience different local environments that are depicted by these different colors. And what we'd like to understand is that how does local adaptation really depend on various factors, including the connectivity between these subpopulations, how local environments are distributed across subpopulations, the genetic basis of adaptation to one's local environment, for example, whether local fitness is determined by a small number of genes or by a very large number of genes. And of course, we might also be interested in things like whether these subpopulations are small enough for random effects, for genetic drift to interfere strongly with selection. And yeah, I mean, in nature, of course, there'll be a whole zoo of other factors, mating system, biotic competition over mates or over resources. But, as a sort of theoretician, one has the luxury of sort of starting out with a very simple model in order to pin down some of these effects more carefully. And so what I'll talk about today is really such a simple model, and I'll try and illustrate some of the nuances that emerge. So the model that I'll consider is as follows. So let's assume that we have a very large number, an effectively infinite number of subpopulations, which are all connected to each other via migration. We assume that the size of each such subpopulation is fixed. So for simplicity, I assume that there are n haploid individuals on each such subpopulation, on each such island. And we also assume soft selection, which basically means that the size of a population is independent of its fitness. And so in each generation, a fraction m of individuals on each island is replaced by migrants who are drawn uniformly from across this whole metapopulation consisting of all these islands. And the fact that they are drawn uniformly, that is with equal probability from any of the other islands, means that there's no concept of near or far. So there isn't a concept of space, but of course there is subdivision in the sense that each such individuals will compete, or their relative fitness will only be determined by other individuals within the same patch within the same island. Then the next point is that we assume that across this entire meta-population, there are two habitats. So a fraction row of islands belong to one habitat, and the remaining fraction 1 minus row belongs to the other habitat. Fitness on each island is determined by the habitat. So we basically assume that fitness depends on L unlinked loci with habitat-dependent fitness effects. And unlinked simply means that these loci segregate independently of each other. That is, they are sufficiently far apart on the genome that they are they are not inherited in a correlated way. And so the model of selection that I'll assume here is a very simple one. I'll assume that if each locus that determines fitness has two possible alleles, then alternative alleles are favored at each locus in the two habitats, independent of what's at each locus, independent of the state of other loci. So basically, if we denote the two alleles by 0 and 1, then at each locus, allele 0 is favored in habitat 1, and at each locus, allele 1 is favored in habitat 2. And for simplicity, I'll also assume that the selective effects of alleles are the same across the different loci. Then relative fitness just depends on the number of locally deleterious alleles that an individual carries. So basically the number of alleles of the wrong habitat, of the other habitat. And fitness is just determined by the selective effect per allele times the number of locally deleterious alleles. So as you see, selection tends to favor different alleles in the two habitats, but of course there's mixing between islands, and this will favor, this will sort of tend to erode genetic differentiation between the two habitats. And since my main focus is on the interplay between migration or mixing and this sort of divergent selection, I'll mostly neglect mutation, except in one special limit, which I'll just come to, where we need mutation in order to maintain polymorphism in the long run. So the special limit, which I'll focus on in one half of the talk, is really this so-called row tending to 0 limit, where one of the habitats is much rarer than the other habitat, such that the state of the more common habitat is largely unaffected by the state of the rare habitat. And for simplicity, we can assume that this common habitat is essentially perfectly adapted. And what we have is essentially unidirectional gene flow from one large habitat, which behaves as if it's some big continent, to a single island. So this is the so-called mainland island scenario, which is just a special limit of this more general model, where you have a very large number of subpopulations and a finite fraction of these belong to one or the other habitat.
Himani, can you explain the dynamics of this one? So what you have is first the migration I do. That means I replace by the fraction m of the migrants, and then I do the drift and selection, or the order doesn't matter? Can you?
No, the order doesn't matter really, because what I'll essentially use is a sort of diffusion approximation, which matches very well with sort of individual based simulations, for example. But yeah, in a simulation, typically I would replace a fraction m of individuals by migrants, then choose parents for based on their relative fitness, then create offspring by free recombination.
And these migrants means the other habitat?
It means that basically you have individuals drawn uniformly from across the entire metapopulation. So some of them will be from islands belonging to the same habitat. So typically a fraction, because a fraction row of islands on in the whole population belong to one habitat. So the migrants will also be from the same habitat, from one of the habitats with probability rho, and the remaining fraction of migrants will be from the other habitat.
So when rho is going to 0, that means most of the migrants would be basically coming from habitat 2. And whereas habitat 2 is not affected because it's smaller size. Okay.
Yeah, and then one can effectively just think of it as a mainland island, asymmetric.
Right.
Focus on a single island and assume that it's receiving migrants from a perfectly adapted mainland. Yeah.
Okay. I have one small question regarding this migration. So
yes.
There are subpopulations, and the population size of each of this subpopulation is constant. And there are infinitely these groups. So how this migration is working, because the amount of individual leaving this particular habitat, there will be some gain in the individuals also. So in that manner, the N is on an average kept to be constant. So how this migration will affect this?
So basically, if you have something like this, you assume that, individuals just, so for example, if a single individual, let's say 2 individuals leave from here, they are replaced by 2 other individuals from somewhere here, from anyone, where you draw these 2 individuals from any of the other islands with equal probability, and so on. So that you maintain a balance in terms of the number of individuals coming in and going out overall.
Okay, and the migrants coming from any of the subgroup has the same fitness, or is there the difference in the
No, so fitness, well, in every island you have 2, so on each island, depending on which habitat that island belongs to, either the 0 or the 1 allele is favored, and the fitness will depend on the number of locally deleterious alleles that an individual carries. So if you come from the other habitat, typically you'll be pre-adapted to that habitat, and you will then suffer lower fitness when you migrate to a different habitat. So just to sort of put it all together, we have a model with the following parameters. We have rho, which is the fraction of islands that belongs to one of the 2 habitats. We have L, which is the number of divergently selected loci, and typically I'll consider L in the tens or hundreds, which means that I'm thinking of really polygenic sort of fitness. Then there is S, M, and 1 by N, which govern respectively the change in allele frequencies due to selection at individual loci, because S was the selective effect per deleterious allele. There's M, which governs the rate of migration, and 1 by N, which governs the rate of drift within local populations. There's also R, which is the rate of recombination between selected loci, and for unlinked loci, of course, R is half. And what we have here is that this recombination is important because it essentially breaks down associations between deleterious alleles, and the faster that recombination is, the faster is this breakdown of associations, or LD, between selected alleles. So the one point, sort of broader point that I'd like to make is that even the analysis of even so simple and caricaturized a model becomes challenging because of 2 reasons. The first is that we have multiple loci under divergent selection. So when a deleterious allele, locally deleterious, locally harmful allele comes into a subpopulation, it doesn't just come in on its own. It comes in as part of a genome in which you have multiple such deleterious alleles. And so the selection that it will experience, at least initially, is not just selection due to its own effect, but really indirect selection due to the fact that it is associated with all these other deleterious alleles. And these sort of statistical associations are known as linkage disequilibria, or LD. And so what we really need to think about is the dynamics, not of single deleterious alleles, but really these multi-locus genotypes which come in and which then are which break apart into smaller chunks of deleterious fragments as they mate, as individuals carrying these multi-locus genotypes mate with other individuals who might be fitter and have offspring who only inherit a fraction of the deleterious alleles. So you need to consider really all of the dynamics of all these multi-locus genotypes. And the other reason that theory is challenging is because if you're thinking of this sort of subdivided patchy population, then individual subpopulations may be small enough for genetic drift, that is for random fluctuations in allele frequencies or even genotype frequencies to be important. And in that case, what we really need to consider is the stochastic dynamics of all these multi-locus genotypes. And this is something we know that this is in general quite a hard thing to do, because most of the methods that we have typically apply to single loci. So for example, things like the diffusion approximation, which is also known as Fokker-Planck equation, this formalism in physics. All of these really are practically applicable only to thinking about allele frequency distributions at one single loci. So most theory actually neglects one or the other aspect of this sort of problem. And what I'll do just soon is to sort of outline how people think about how this sort of dynamics, the approaches which ignore one or the other of these complications, and then sort of outline a heuristic way of combining these approximations. So to sort of take also a broader biological view, one reason why this...
Himani, question?
Yes.
Just wanted to clarify. So then are you going to assume S to be much, much smaller than R or something, so that one can think of at least quasi linkage?
No, I won't assume quasi linkage equilibrium actually, but R is of course half. So we have unlinked loci. And
okay, yes, okay. Yeah. So R is pretty large, yes.
S, I'll maybe it will be clearer as I come to it, because I'll go through it in some detail. Yeah. Okay, so yeah, so sort of to also take a more broad biological view of this question, when we think about speciation and the buildup of this sort of genome-wide reduction in gene flow between populations, this process often, though not always, requires basically coupling between these different genetic incompatibilities or different genes that contribute to, which are under some form of divergent selection or which contribute to loss of function in hybrids. So it's not enough to have one or two such alleles. What we require is the buildup of associations, or the fact that individuals who carry one divergent allele should also carry multiple such alleles, so that there is so that any location on the genome is somehow linked to one of these incompatibilities. And this sort of process of coupling is quite essential in the buildup of speciation in many cases. And these sort of associations also are known to result in these sharp threshold effects, where there are these sort of cooperative transitions where there's a sharp change in, let's say, divergence either at some point in time or beyond a certain level of migration. So this is something that people have spent a lot of time thinking about. But the sort of point of view that I want to take here is to ask, how does drift really, genetic drift, really interfere with this sort of these sort of sharp thresholds? So, of course, one effect of genetic drift is that direct selection against deleterious alleles can be weaker when n is small. But the other effect also is that there can be some kind of interference or negative LD due to drift. So, and this sort of counters these cooperative changes, which are because of sort of concerted changes across multiple loci. So, how do we, how do we think about these mechanisms which of coupling in populations that are patchy, which is, the case for most natural populations? Yeah, there's a question.
Yeah, hi, Himani. So could you please explain this negative LD interference a bit more? I'm not very familiar.
Yeah, I mean, this is a whole big topic actually, so I'm not sure to what extent I'll manage, but basically what this means is that, so when you have in small populations, you tend to have, so basically what will happen in small populations is that if you, so imagine that you have genomes with multiple positive alleles, which are on the same genome, these will rapidly increase and fix in the population. If on the other hand, you have a situation where there are different individuals carrying different deleterious alleles, so deleterious alleles are different different locations, then on the genome, then their fitness may be roughly similar, but so this sort of makes the competition between these individuals weaker in some sense, which causes these configurations where you have where different deleterious alleles are on different genomes to sort of persist in the population for a long time. And of course, if there is a lot of recombination, then you keep shuffling and you keep bringing together these alleles rapidly onto the same genome, and then they can be eliminated more efficiently. But if recombination is also limited, then you have these configurations where you have individuals carrying different deleterious alleles on different locations on the genome to sort of persist for long times. And this happens more in smaller populations. But yeah, I mean, so the broad point that I guess I'm trying to make here is that it's not obvious how some of these sort of more cooperative effects where groups of deleterious alleles are sort of eliminated in a cooperative manner when they come in and as part of these immigrant genomes, how this process occurs in these patchy populations is sort of is not clear, and that's something we hope to understand here.
Okay. Thank you.
So I'll briefly indicate the sort of approximations that I think are helpful in thinking about this problem, and then I'll apply it to the mainland island model, and then finally this infinite island model with two habitats. And this is sort of based, whatever I'm talking about is based on this preprint, which is on bioRxiv. So let's maybe start with just to see how much time. Okay, so let's start with the sort of approximations that are useful here, which help us think about this problem. And I'll illustrate these in the context of the mainland island model, and where we are just focusing on one island, which is receiving migrants from this large mainland, but the approximations are also very similar, conceptually similar approximations also apply for the infinite island model. So let's first start with approximations that neglect LD. So this or neglect associations between these different loci. So these basically apply when recombination, which is order half, is much faster than all other processes. And so more specifically, recombination is faster than LS, which governs the strength of selection against maximally unfit genotypes, basically genotypes that carry locally deleterious alleles at each of the L divergently selected loci. So in this case, individuals who come in carrying, individuals from the other habitat that come in carrying these locally deleterious alleles at all L loci are rapidly broken apart by recombination faster than they are acted upon by selection. And so one can essentially ignore the fact that, the migrants that come in have all these multiple deleterious alleles. And in this case, one can essentially assume that loci evolve independently on the time scales at which we have selection and migration and so on. And for independently evolving loci, we know what the allele frequency distribution should look like under migration, mutation, selection, drift balance. And this is given by Wright's famous formula, which you might have come across. And this is basically, so this gives you the distribution of allele frequencies in the population. The important thing here is that the distribution doesn't depend on N, M, and S separately, but really on these scaled parameters, NM, NMu, and NS. The other thing is also that this distribution is basically U-shaped in small populations, or more specifically when NM is small, because what that means is that at any individual locus, you are close to fixing one or the other allele. So allele frequencies are close to 0 or 1. And of course, for larger migration rates, or in larger populations, you might have more intermediate frequency polymorphisms. So this is...
Yeah, I haven't thought about this distribution with migration. So can you just help me a bit here? So I'm just trying to understand why only P going to 0, K, part more affected by migration. Can you give some insight into that? So we understand without migration, yes, it's a U-shaped function.
No, so if you had no selection, then also just with migration, it would be a U-shaped function as well.
Okay.
Sorry, in this case, yes, sorry. So that's only because, on the mainland, we are fixed for one allele. Sorry, that I think I was saying something else earlier. So migration basically introduces only, so if there was only migration, then you would essentially fix the mainland allele if there's unidirectional migration.
Yes. Let me see again. Okay, first of all, 2mu is less than 1. So therefore you have U-shaped distribution and slightly skewed because of selection. Okay, now there you have also migration, which affects small p. And as you increase migration, it becomes different way it goes. Instead of diverging, it goes to the frequency becomes 0. So I think I still don't understand that qualitative behavior.
No, so if you only had migration, or when migration is much stronger than everything else, because this 2nm term comes only here, not here, yes, because the mainland is assumed to be fixed for the locally beneficial allele on the mainland. So when you have only migration, you're constantly driving the allele with asymmetric migration, one-way migration, you're constantly driving the allele frequency on the island towards the allele frequency on the mainland, which is p equals 1. So at high migration, you're now, as migration becomes higher, this will become even more peaked at p equals 1.
Okay, fine. Yeah.
So it's just a consequence of the asymmetric migration and the fact that the allele is fixed on the mainland. Okay. So this is, yeah.
Sorry. So in your model, the migration will be symmetric.
So this is the case, the first, right now I'm talking about the mainland island case, where you have a large, well-adapted mainland, or one habitat is large and well-adapted. So effectively, because most of the migrants come in from this large mainland, its frequency is fixed, and you can think of this as just asymmetric migration from a large, perfectly adapted mainland to an island. But in general, of course, I have a situation where each of these is connected to each of these other islands by migration.
Okay, thanks.
Yeah. So this is all to do. So this is just a single locus picture where we ignore associations, but account for the effects of genetic drift. We could also do the other thing, which is to assume that drift is weaker than all other processes. So basically n is very large, or 1 by n is smaller than everything else. And in that case, you have deterministic equations for genotype frequencies. In these equations, I'm no longer going to assume that recombination is much stronger than net selection, because, this would, we don't need to assume that. We can basically just write deterministic equations for genotype frequencies. So basically, equations for P1, P2 up till PL, where P1 is the frequency of genotypes that carry just one locally deleterious allele, and PL the frequency of genotypes that carry L locally deleterious alleles. And one can write down these equations under migration, selection, recombination, balance. And these are in general rather complicated, and you can't really do much with them. But if one assumes that these deleterious genotypes are rare, they're not too abundant in the population, then one can essentially write down linear equations and solve these, and obtain, use these to basically obtain the average deleterious allele frequency in the population. And it has this complicated form, which is not so important, but what it becomes is that this average deterministic deleterious allele frequency for when you have small s, basically weakly, when each individual deleterious allele is weakly selected and there are a large number of them, then this takes on this rather simple form, which is that it's m by s times this factor. Now, m by s is just the deleterious allele frequency, and if you had just a single locus under migration, selection, balance. And what we are saying is that the fact of that it's not a single deleterious local, but what you have is these multi-locus deleterious genotypes that are coming in. It's as if this reduces the effective migration rate of any such allele by a factor, which is this e to the minus 2ls. And this also has a nice interpretation. So what one can show, but I won't go into it so much right now, but what has also been shown in the past is that this is basically this e to the minus 2ls is just the reproductive value, which is basically the long-term genetic contribution of an individual carrying L deleterious alleles of S, a disadvantage per allele being S. It's the long-term relative contribution of such an individual to the gene pool of the population into which it enters. And so what effectively is happening is that whenever you have such a deleterious, whenever one has this sort of multi-locus genotypes that are being exchanged, it's as if the effective rate of migration of each individual allele within this large multi-locus genotype is reduced by a factor equal to the long-term reproductive value of individuals, which just depends on LS, which is the net selective disadvantage of the multi-locus genotype. And I've done all this for selected alleles, but one can sort of come up with analogous calculations for neutral alleles. So this picture again is just neglecting drift and assuming that deleterious alleles are rare or deleterious genotypes are rare. So now what one would like to do really is to combine these two approximations and ask, can we sort of describe what happens if, firstly, if the rate of drift is comparable to migration and selection, at least at individual loci, such that drift interferes, can interfere with selection at the level of individual alleles. And also if we cannot really ignore associations. In other words, if recombination is comparable to net selection against genotypes, and remember that recombination is just half here. So if it's comparable or even actually lower than weaker than net selection against genotypes, so that interference or association between alleles is also important. So drift is also important, and associations are also important. Of course, I will still assume that recombination is, in this case, by definition, because it's unlinked loci, so recombination is order 1. But I'm not, as Kavita was asking earlier, I'm not going to assume that selection and recombination are comparable at, recombination is still, much faster than selection at the level of individual alleles. And this sort of makes sense if we assume that we are thinking really about polygenic architectures of hybrid dysfunction, where individual, or polygenic architectures of local adaptation, where individual alleles contributing to local adaptation are typically not that strong, even though their net effect overall could be quite strong. So in this case, again, what we see is that there is nevertheless a natural separation of time scales that comes up because, basically because LS, which governs net selection against multilocus genotypes, is much higher than S, as long as, we have many such deleterious alleles. And so what one can also see from here is that if drift affects single deleterious alleles, that is, if 1 by n is comparable to S, then it will necessarily be much weaker than genotypes, than selection on multilocus genotypes which carry many such deleterious alleles, or which carry L such deleterious alleles. And so this sort of suggests that maybe we could stitch together a deterministic description of multilocus genotypes, the dynamics of multilocus genotypes, with a stochastic description of the dynamics of single locus, of individual loci. And one sort of relatively simple-minded way of doing this, or rather ad hoc way of doing this, is to assume that allele frequencies at individual loci are still given by the diffusion approximation, but with an appropriately chosen effective migration rate rather than just a raw migration rate. And then, of course, the question becomes, but what is this effective migration rate? Is it just this? Is it just reduced by a factor which depends on the net selective disadvantage of migrants? And of course, this makes sense if, migrants come in carrying L locally deleterious alleles. But we would also like our sort of theory to be able to predict when adaptive divergence is lost between habitats. In other words, when fitness differences between the two habitats actually shrink because of the fact that you are close to fixing the same allele in both habitats. So we'd like to get at the thresholds at which local adaptation is actually lost. So it turns out that so one can sort of argue that the effective migration rate in that case doesn't depend on the number of divergently selected loci, but actually the number of loci that actually contribute to fitness differences, or the fact that migrants will have lower relative fitness when they migrate from one habitat to the other. And this is not really the number of divergently selected loci, but actually the number of loci that contribute to fitness differences. In other words, the number of loci at which the island is fixed for the locally beneficial allele, while the mainland is fixed for the locally deleterious allele. For the mainland is, the mainland is always fixed for the locally beneficial allele. So it's the number of loci at which the island is able to withstand gene flow, and it's the number of loci at which it's fixed for the beneficial allele that is locally beneficial on the island. So one thing to, of course, see is that, and this is where the U-shaped distributions come in, that when we have, so imagine that our expected frequency of the locally beneficial allele is 0.5 on the island. This doesn't mean that at each individual locus there's a 50:50 polymorphism. What it means is that instead half the loci are close to fixing the locally beneficial allele, while the other half are close to fixing the locally deleterious allele. So this tells us that the number of nearly fixed differences between the mainland and the island is just L times the expected frequency of the locally beneficial allele, which is 1 minus the expected frequency of the locally deleterious allele. So now, finally, we have a allele frequency distribution which depends on the effective migration rate instead of the raw migration rate. And this effective migration rate itself depends on the expected allele frequency. So what we'd like to do is to essentially obtain this self-consistently by numerically integrating it.
Himani? Yeah. So, I mean, like, one constraint is simply that the psi being a distribution should be normalized to 1. So won't that say something about ME?
No, I mean, one can basically, so one can obtain expected P as a function of.
On both sides, suppose I find expectation of P, you're saying?
Yeah, exactly. So, and then you obtain it as a expected P. So let's call this, so you obtain it by.
I think I get it,
yeah. Sorry, I should have had a figure to describe it. Yeah.
Thanks.
Okay. So this is finally
could
the
you explain this? Could you explain this extra term 1 minus EP? Because we just saw, I was guessing that M will be simply, ME will be ME to the power minus 2 SL, like we had in the deterministic case. But so, could you explain this extra term?
Because, so if you remember, what matters here is that, so when I assume that deleterious alleles are rare, what I effectively end up assuming is that the mean fitness of the population is hardly affected by the fact that there are some of these migrants whose fitness is very low, because they're so rare that they don't affect the mean fitness so much. And their relative fitness, so the mean fitness is essentially 1, and the relative fitness of these migrants is e to the minus 2 ls. And what this ends up, and what I'm trying to do here is to not just constrain myself to situations where the deleterious frequency is low enough, but actually to situations where the mean fitness is also affected by the presence of migrants who may actually be quite abundant for example, at higher migration rates at which local adaptation is about to be lost. And in that case, what will determine the relative fitness or the relative reproductive value or contribution of these individuals is not 2SL, but really 2SL relative to mean fitness, or in other words, the number of deleterious alleles that are number of loci at which the mainland and island populations differ in frequency. And so, for example, if expected P, which is the frequency of the locally deleterious allele, is 1, then the mainland and island populations are all essentially fixed for the mainland allele, and then the relative fitness of individuals coming in is, there is no difference in relative fitness between migrants and residents, because there is hardly any genetic variation.
Okay, yeah, thanks.
Yeah. So, so let me then just try and indicate to you how these approximations work. So I'll do this first for the mainland island model, and if there's time, I'll briefly touch upon the infinite island model, where I also think about neutral divergence in FST and so on. But let's first look at the mainland island model. So here I am showing, the plot shows the expected frequency of the locally favored allele on the mainland versus the average number of migrants per generation for different numbers of divergently selected alleles. So you have L equals 1, that's the brown, then L equals 10, and up till 80 divergently selected loci. And the symbols here represent the results of full individual-based simulations, and the lines represent the results of theory. So one thing that, of course, we see here is that the theory and the simulations match remarkably well, which suggests that this simple heuristic that we have of trying to capture the effects of multilocus LD via just the single effective migration rate that only depends on the average fitness difference between the mainland and island, that works quite well and is able to capture the sort of effects of LD at the level of effects of LD on allele frequencies at individual loci.
So that it's at NM equal to around 0.9 for the blue curve. I guess it's not some sharp drop. It's just you've just joined the points.
No, it is quite a sharp drop. I mean, that's yeah, because so this is all. I mean, this is the theory. The simulations I don't have for so much, but the theory is, so this is, I mean, this bit is joined, but actually it's quite a sharp drop. And this is generally a feature of whenever we have LD, that is whenever your LS is high, then you have these sharp thresholds or tipping points for loss of local adaptation. And this sort of sharp, of course, this would be less sharp if, for example, the effect sizes were very different across loci, but with equal effects, you do have these sharp effects. And even with unequal effects, actually, depending on the distribution, you do see these sharp thresholds. And this has to do with the fact that beyond a critical migration rate, when there are large numbers of divergently selected loci, then once small changes in allele frequencies at very many loci have a large effect on a large aggregate effect on the effective migration rate of individual alleles, because that depends on L times changes. So there is, and then the effective migration rate per allele reduces, which increases the frequency of the the effective migration rate of per allele increases, which increases the frequency of the locally deleterious allele, which further increases the effective migration rate, because it depends not just on allele frequencies at individual loci, but on the aggregate effects across all these loci. And there's a positive feedback, and that results in these sharp thresholds. And this is something that people have known, for example, in large populations, that there are these sharp threshold effects. So there's work by Nick in the 1980s, for example. And it's a general feature whenever LD-driven transitions are occurring. And that's why you only see it for large L, not for small L.
I see. Okay, thank you.
Yeah. And I mean, the other thing is that, of course, the frequency of the locally favored allele increases per locus when you have more such loci. And that's again to do with the protective effects of LD, that when you have individual alleles coming in as part of these multi-locus genotypes, then they have this selective disadvantage in the initial first few generations, or this reduced effective migration rate, which is due to all these other alleles. So you can protect the locally favorable allele and have higher frequencies at when there are many more loci.
One more thing, Himani, just because I don't know this results, so just want to understand. So suppose L goes to infinity, so that does this blue curve, does it become more and more like a step function?
Yeah. So it depends on how you take L to infinity.
If I keep increasing L, so I'm just trying to see, because that shape is much easier.
Yeah. So then it does become more like a step function. But it also depends. So, I mean, taking L to
but LS should remain higher.
LS should remain. So ideally, one would, what we know, LS is about. LS tells us that what's the fitness of F1s. So this would not, if you take L to infinity, then it doesn't make sense to keep S constant, because then you would have F1s or hybrids essentially having zero fitness. So one way of doing this is to keep LS, which governs fitness of hybrids, to be the same, and actually take L tending to infinity, S tending to zero limit, where you're thinking that the same level of hybrid or same level of disadvantage is due to many more loci of weaker effects. And then all of this reaches a certain limit, which is different. Yeah. Then it doesn't reach a step function necessarily.
Yeah, but that would be a non-trivial limit. If you take a large S small, such that this is so. Now I was just thinking of really a limiting case. So if I just take L to be really infinity, I expect a step. I mean, does it become a step function? So that's all.
No, if you take L to infinity, then LS will also become large, and then actually this curve shifts. And I'll maybe come to that. I probably won't have time to come. Yeah, because so right now what we're seeing is that LS, this curve is not shifting, it's just becoming sharper. But beyond a critical level of LS, the curve also shifts. That is, you have
different critical NMs?
Different critical NM.
Okay.
So yeah. And one can predict what is the level of LS, or one can at least get an approximation for what is that level of LS beyond which there is shift, as opposed to just sharpening of these thresholds.
Okay, thank you.
Yeah. I'll skip this because I guess I'm running out of time. Let's just look at, so that was just indicating the effects of increasing the number of loci while keeping NS constant. Now I'll try and ask a slightly different question, which is that suppose we have a certain number of loci, let's say 40 loci here, each of selective effect 0.02, such that LS is 0.8. And we could ask, what is the expected frequency of the locally favored allele, or how does local adaptation depend on migration relative to selection in populations of different size? So the different colors here indicate populations of different size. We have NS, so ranging from N equals 50 to N equals 800 here, the blue curve. And the symbols as before indicate simulation results. The solid lines indicate what one predicts with this whole approximation that I outlined, which accounts for both LD and drift. And the dashed lines for each color represent what one would predict if one ignored LD. So basically just thinking about Wright's formula, single locus linkage equilibrium analysis, but of course accounting for drift. And the dotted line here, the black one, indicates the prediction if there was no drift. So of course, as before, the theory and the simulations agree quite well. And so what we see is that drift can strongly reduce the migration thresholds for local adaptation. Essentially, in smaller populations, you lose local adaptation at much lower values of M by S, and in larger populations, of course, it's higher. And and this, yeah, so there is, and of course, we, as you see that with increasing N, you converge towards this deterministic prediction, which neglects drift altogether. The other thing sort of we can see is that LD has a much more significant effect in larger populations. That is, if you look at this, the actual levels of divergence as opposed to what you would predict if you ignored LD, there's a much starker difference for the blue curve as opposed to the brown curve. So what we see is that this cooperative elimination of alleles is much more effective in larger populations than in smaller populations, and that this is quite well predicted by this heuristic of using effective migration rates. So not only can one predict, the expected allele frequencies, one can actually predict the whole site frequency spectrum for different values of for different migration rates. And I won't go into details because of, I guess, the time limit, but it's interesting that it seems to predict, so the lines, which are the theory, seem to predict the simulation results quite well, even when the allele frequency distributions or the site frequency spectrum is not U-shaped actually. Sorry, not site frequency spectrum, because that would be if one took a small sample, but when the allele frequency distribution is not U-shaped, even then the theory is reasonably accurate. I guess I should try and wind up, so I won't really go into this and the case of the two habitat model, except to say that this sort of extends quite well, and one can ask, what are the values, how do critical migration thresholds depend on selective disadvantage? So this is what I was talking about earlier, that they don't change for a certain value of LS, but above a certain value of LS, not only are the thresholds sharper, but they also shift. And one can also predict neutral divergence, that is FST, based on this sort of formalism. And I'll just summarize that. So the key point here is that when local adaptation is polygenic, there is a natural separation of time scales that emerges. Drift must be comparable to selection per allele if local adaptation is to be maintained over at all. But that necessarily means that drift is much weaker than selection on multilocus genotypes, which carry many such deleterious alleles. And this allows us to splice a sort of deterministic description of LD based on these effective migration rates with a stochastic description of allele frequencies based on the diffusion approximation. And this rather simple-minded way of doing it, where we just assume that it's as if allele frequency distributions are well predicted by the single locus approximations, but with an effective migration rate that depends on the fitness difference, average fitness difference between the two populations between which migration occurs. This sort of heuristic actually works surprisingly well. LD between divergently selected alleles results in sharper thresholds as well as shifted thresholds, which I couldn't go into. This emerges from the infinite island case, where you have a finite fraction of islands in the two habitats. And there were also sort of interesting predictions about neutral FST, which emerge from this sort of analysis. So maybe I'll stop for now, and yeah, if there are questions, I could take those.
Any questions? Okay. Yeah, I don't see any questions. Yeah, I don't know your first point, and just I haven't, of course, thought about it. But if I don't have a migration, just well mixed population in a single island and so on, these other models, with stabilizing selection, for example, do you think one could splice the deterministic approximations and the stochastic one? I mean, it's a sort of vague question. I'm not sure what I want to substitute as an effective thing, but do you think this kind of idea is pushable in those kind of problems?
Yeah, I mean, I think so. What is important here is that there is there's a separation of time scales, and so yeah, I should say that. So the short answer is I don't know, because one of the big surprises with this is that it works remarkably well across a whole range of parameters, but I don't have a formal justification for why it works. It's not some sort of perturbative expansion that you can say, okay, it comes from, it should fail when this happens or that happens and so on. So it's very hard to give a sense of when it fails. I mean, one thing that I was trying to do with this is to look at genotype frequencies and say that, is it that you have, this sort of, so this is the genotype frequency where the number X is of the deleterious alleles in the genotype, and this is, genotype frequency, the frequency of genotypes carrying X alleles. So some intuition that I had initially was that maybe, there is this deterministic part of the genotype frequency distribution, which is for the large, more deleterious genotypes. And then this is the part that this is the curve that you get if you assume just LE, and there are these two distinct regimes or two distinct parts of the genotype frequency distribution. And I mean, this sort of intuition, of course, holds for very weak migration, where, you have such genotypes come in, then they disintegrate, and then before the next one comes in, there were these two distinct regimes. So the dashed line is the deterministic genotype frequency distribution, and that solid line is what you would get under assuming LE with a certain allele frequency. But then it doesn't work, you don't really see this for larger migration rates, whereas the actual prediction for
you are measuring some sort of an average quantity, right? So maybe if some distributions are not working, but because what you plotted earlier was some expected quantities, right?
No, this is the genotype frequency.
This one is a distribution.
Yes.
But what you showed us earlier is some expectations.
Well, the point is that it seems to work at the level of individual loci. So even the allele frequency distribution, not just the expectation, but the actual distribution here is also quite well predicted. So this is the allele frequency distribution. So, I mean, I think maybe the way to go is to think more in terms of reproductive values that which, there's this whole set of literature by Fisher, but also then again by Alison Etheridge and Nick about, what is the long-term contribution of a genotype to the genetic diversity in a population, or in other words, if you have a neutral allele carried by a genotype, what's the probability that it will be part of the population in the long run and not just be weeded out by selection? So, I mean, I would maybe explore, and that doesn't have anything to do with migration, actually. That's just about, you can think about it in a single population. So one possibility is to somehow, I don't know whether one can think of deterministic reproductive values and incorporate them in some way into a stochastic description, because ultimately here, this factor is really just the reproductive value of genotypes. But yeah, I also don't have a good answer, because I think it would be interesting to actually pin down a proper justification, if any, for this sort of approximation.
Yeah, but it's nice to see if one can sort of couple these things, because usually full-blown stochastic models, one just can't do it. For this polygenic problems. So we are still stuck with our old stabilizing selection drift problem. So that's what I was sort of thinking. But okay, I think I'll probably look at your paper and think a bit more. Yeah. Okay, so I don't see any more questions. Okay, so thank you very much, Himani, for joining.
Thanks.
Okay. Good luck.
Thanks, Himani.
Thanks.
Okay. Bye-bye.
Bye.